We're really excited and fortunate today to have a good friend of mine and collaborator for a long time, Dr. Brian Roberts. Brian is the executive director now at LUMCON, but before that, he did his bachelor's degree at William and Mary, master's at Woods Hole and Boston University, a PhD at Cornell, followed by a postdoc at Oak Ridge National Lab. So he's a very good background before going to LUMCON, and he has ascended the ladder to executive director there. So I've asked him to talk just a little bit about LUMCON and all the facilities and new improvements that they've done at LUMCON recently and the direction they're going, in addition to some of his own research. And I can attest that Brian's research is very much ecosystem based. You know, he's often labeled as a biogeochemist, but I've worked with him on everything from community ecology to animal behavior. So excited to have you here today, Brian. So take it away.
Thank you, Charlie, and thank you everybody for coming. So as Charlie said, he asked me to give a little bit of overview of LUMCON. So strap yourself in, I got a few slides. So LUMCON's mission is to promote, facilitate, and conduct research and education collaborations among Louisiana's universities in marine and coastal sciences. Our vision is that our coasts and oceans are revitalized through innovation, collaboration, community action. We kind of have this mantra of connect, enrich, transform. Connect is to enhance research, collaboration, exchange by linking the consortium through stronger partnerships. Enrich is to give back to the community through education outreach initiatives. Transform is to impact science and conservation in meaningful and profound ways. And I throw this up because you guys are probably one of the only places in the country that can actually relate to this drawing that I have here. And the fact that we're a marine lab, but we're also a consortium. And so the LUMCON consortium consists of us and to our resources and programs, but then all of the systems in the state. So all the schools in the LSU system, in the University of Louisiana system, the Southern University system, the community and technical college system, as well as private colleges and universities, which does make us a bit unique in that. So in total, we have somewhere between 35 and 40 universities that are engaged in the consortium. LUMCON is a program of the Board of Regents. So I report directly to the Commissioner of Higher Education. We have 5 main components, the way that I look at LUMCON, to achieve our mission. We have all our facilities and resources. We have our research and science programs, education outreach programs. We have the state's research vessel and dive operations. And then the Barataria Terrebonne National Estuary Program falls under our umbrella as well. So we're this unique location for research and education, access to the largest wetlands. Our property actually extends from the road here over the Home and Navigation Canal. So these wetlands are all part of LUMCON itself. We currently have 7 faculty in residence with an 8th one who is starting at our new campus I'll talk about in a moment. He began earlier this year, and we're currently recruiting 3 additional faculty. Many of these people are folks that you guys are familiar with. Nancy Rabelais' appointment is now officially at LSU, but she still maintains most of her lab at LUMCON. And then we have several other faculty. I mentioned we're recruiting some additional ones. Our Marine Center has dormitory rooms and apartments that can provide housing for around 80 people. We have a multi-use auditorium, not that much unlike this one. In fact, and then we have research laboratories and saltwater facilities. This is a marsh mesocosm facility that we built during the Montgomery era, looking at impacts of oiling on a larger scale experimental systems. We have a lot of wet lab facilities, things that you guys are probably familiar with seeing. We have a racetrack flume. We have an extensive library, 3D printer labs. We also are the state's Marine Synthesis Center, so we have working groups and different types of meetings that we support. And we have a short-term visitor program that part of our funds to cover people to come for short durations of times to spend time at LUMCON. Our education outreach is something that's similar to what you guys do here. There's a lot of parallels our marine education program invests in communities to enriching relevant place-based and skill-based experiences for all learners among Louisiana's diverse citizenry, including population underrepresented and underserved marine science. Purpose is to support and empower current and future marine scientists and ocean literate citizens. And these are programs ranging from pre-K to gray. So you have the pre-K through 12, our university education programs, our community education programs. And we have, obviously, our busiest time is the summer. That's here. We also have the state's research fleet. So our flagship is the RV Pelican. We also have the RV Acadiana, which is comparable in size to some of the boats that you guys have here. And then we have a diversity of small boats as well. The Pelican is approaching its 40-year birthday in 2025. And so we're well on the way for its replacement. Actually, today, the requests for the qualifications are actually due today. So we should know who's actually trying to bid on building this boat. And we expect the notice to proceed around the turn of the year and hoping for an 18-month, but we all know that's never going to happen, delivery time. We're also, the National Science Foundation is building 3 research class research vessels, which are a larger size here, 199 feet. They're all being built in Houma. The third one out of the shipyard is going to be jointly operated by ourselves with the boat operations and University of Southern Mississippi for the technical and marine services. That will be mainly used in the Gulf of Mexico, depending upon the research. These dates of when they're going to come off the yard keep sliding. So this is now May of 2025, when we're expecting to have the Mason. So Charlie said we're doing a lot of new things. LUMCON is actually expanding into a maritime campus closer to my house, which would be an easier commute than going down to Coventry every day. This is land that was originally given to the community college system in Louisiana. They occupy this building over here. And then this plot over here on the left side of the diagram is one that has been given to LUMCON. So the first is our Blue Works facility I'll talk about, then the boat slip and bulkheading that can accommodate the RCRV as one of its 2 dual home ports, and then a new marine operations center, as well as trying to extend the bulkheading along the property. This is what the exterior of our new building looks like. It's 27,000 square feet in total, and we are doing a ribbon cutting on this in early December. The boat slip and bulkhead, that construction began in September and should be completed by next summer. And then the Marine Operations Center, a couple more sketches of what this should be looking like. Right now, the design's been completed and we have enough money to start the build. We need the additional money moved from the non-real money to real money in this next session. So we're hoping to have that built before the Mason comes online. And then the latest news is that Louisiana is actually the last coastal state to get a National Estuarine Research Reserve. The governor accepted the nomination of the Atchafalaya River Basin to be our reserve for Louisiana. And in that same press release, it also announced that LUMCON has been chosen to be the state agency to manage the NER. And so we're really excited about that opportunity to bring the power of the consortium and the collaborations behind that and filling in this important gap in the National Estuarine Research Reserve system. And it will be unique in that system because this is a large river delta, right? Unlike any of the other systems in there, this is a really large river. So we're really excited about that. So what that means is that when I look at LUMCON in our future, it's really, we've added this 6th component to it as our arms of LUMCON. So that's a very brief overview. I know it's a lot of slides and longer than probably I should have spent on it, but hopefully it gave you a little insight on what we're doing at LUMCON and where we're headed. And I'll be happy to take questions about LUMCON in general after the presentation. When I was asked to give a research presentation, I decided I wanted to try to give an update on some of the work we've been doing through funding through the Bureau of Ocean Energy Management. This is work that we're doing out at Ship Shoal off Louisiana coast and looking at the biological responses to dredging and showing how these responses depend upon the dredging depth and the time since dredging completion. So this is a large collaborative project. I'll be talking about this first 3-year phase of the work in which Jimmy Nelson at University of Louisiana Lafayette has been the lead PI. We actually just recently been awarded 3 more years to extend the funding, in which case it's shifting to me as the lead PI, as Jimmy's now moved on to Georgia. He's still part of the project, but it needs to be based in Louisiana. So Louisiana has a series of these sand shoals offshore. In Louisiana, our sand shoals are all relic barrier islands from when the delta had a different configuration. And as the delta shifts, it's no longer getting the sand supply to maintain the barrier islands, and then gradually they sink. And these are really important habitats. An area that's dominated by in the summer is large area of coastal hypoxia, so it tends to be a refuge from that. We know from previous work that it supports high concentrations of benthic algae, but nobody's ever measured the primary production on there to understand how much carbon is actually available to the rest of the food web. It's an important spawning ground, and it's obviously a very large sand resource, which is the reason why BOEM is interested in this. Louisiana has lost, almost 5,000 square kilometers of land over the past century from a variety of different factors, as subsidence and sea level rise, among others. And if you look at the projections, if we don't actually do any restoration efforts, what will our land look like? And we'll see that, Cocodrie and the Marine Lab will not be on land. It's a good thing we're up high in the air, you'll be able to access it by boat from underneath. Trying to put that off as long as possible. So really what we were tasked to do in this project is looking at Ship Shoal. This is area over here, highlighted just off of Terrebonne Bay. It was well studied prior to dredging. There was a dredging project that was completed in 2017. It's this Caminada area over here in the far east. This Timbalier Terrebonne pit is one that was dredged in spring of 2021 through June of 2022. And then we have undredged reference areas. And so the overarching project goals, we were commissioned to determine the ecosystem effects of the dredging, determine if there are functional differences in control sites and the borrow sites, and if so, investigate those differences to correlate with biophysical differences, grain size, flow characteristics, etc. This is a little bit closer view of the area, and this is a modified before-after control impact design. We were actually originally scheduled to start sampling in 2020 in March. Something happened then, and it kind of delayed the process. It did not delay, unfortunately, the start of dredging. So it meant that instead of having an entire year of pre-dredge data, we had a season of pre-dredge data for the new pit. We have 3 regions. The reference areas, this new pit, Terrebonne or Timbalier, that's where ultimately the sand was being deposited. And then Caminada is the relic pit. We have 3 sites within each region that are 500 meters by 500 meters. And we sample in 3 seasons: spring, which is March or April, summer, which is mostly in June. Sometimes they carry into the first couple of days of July, depending upon weather, and then fall, October, November. So in terms of the primary production aspect of things, which is primarily what I'm going to be talking about, we looked at the primary production in the water column through CTD collections, and then box cores for the benthic collections. For the water column productivity, we basically did photosynthesis-irradiance curves. We incubate different BOD bottles at a variety of different light levels, and look at the change in dissolved oxygen at each one of those levels and apply a PI curve to the in situ light to determine integrated GPP. I'm not really going to talk about that because I want to talk about the integrated responses in the benthos today. Well, lots of people still being emitted. So the benthic primary production part of this has been a central focus of my master's student, Alex Dowes, who's getting his master's through University of Louisiana Lafayette. Actually just turned in his thesis draft to his committee on Monday and defends on the 30th. So we have, just to show you a little bit about the core incubations. Basically, we collect the cores intact, we bring them back to LUMCON, we siphon off the water, and we put site salinity water that we know the nutrient status of in these cores. We run them in these incubations with these full light spectrum panels up at the top. and each one has a continuous log of dissolved oxygen concentration over time. And this is taking a little longer than I hope, but you can see the light bank can get blinded here really quickly, and what it looks like over time. So what we did is we do incubations in the dark plus five light levels. So we do this completely in the dark. That's to get you your respiration rate. Then we have it with the light on with shade cloth, and the light on its lowest setting without the shade cloth, and then increasing light levels to develop a curve. What this looks like is you're looking at the change in dissolved oxygen over 3 to 4 hours, and you're looking at the linear portion of oxygen change over each incubation. So each of these lines is the change in light treatment. And so what I have in the bottom here are some summary things that show what the slope of the line is and how tight a fit it is in the dark, and then each one of the light levels that we used in this pilot study originally here. What that does is you translate it, you can then calculate the GPP rate for each light level, and you fit essentially Michaelis-Menten curve to it to understand and model what productivity is at any given light level. So when we go out and we collect cores from each one of these sub-sites within each region, we do the incubations and we develop PI curves. So for each one of these are the cores that met our criteria in terms of quality of fit. So we have, in this particular season, from this site, we have 5 cores that had incubations. And so for each one of these PI curves, we calculated a rate of GPP. But for what the illustration I want to show you moving forward, I'm showing you a seasonal composite. This is sort of what the average is of each of them. But we calculated the rate on individual cores. But you'll see in a minute how busy it would get if I tried to do this without every single one of these lines on here. So I want to show you what the seasonal patterns in GPP potential, which is what these PI curves are. So this is in the reference area. This is the spring. We tend to have higher potentials in the summer, and the fall tends up to fall is somewhat intermediate between these. But we also see some degree of interannual variability. For example, our PI curve is very different in the summer of 2021 than it was in the summer of 2022. The big difference between those is the fact that Gulf circulation led to increased clarity and lower nutrient water in the summer of 2022. Any of you who've been off the Louisiana shelf, you know that far off, only 15 kilometers, that the water is brown. And usually I lose sight of the box core by the time it gets down a body length from me. In the summer of 2022, I actually could see the box core on the bottom at 12 meters deep. So this is a real increase in our water clarity. And then the fall PI curves as well. The real question here is it's important because we don't have estimates what productivity is on the shoal to begin with. So it's important to look at the reference, but we're also really tasked with understanding how dredging impacts this. So this is the same reference that I showed you before. And then this is what the mature pit looks like. So there's a pretty consistent pattern of having lower potential rates throughout all seasons in terms of the GPP potential. And then the new pit tends to be largely intermediate in its response. So that tells you about the potential at any given light level. But you also have to remember when you dredge, you're making the system deeper. So what's happening is with this light attenuation, you have different amounts of light at each of these depths. So 8 meters is the average depth for the reference areas. The new pit was only dredged to 10 meters, whereas the relic pit or mature pit was dredged to 12 meters. So that means that you're actually sliding to the left down these curves. So not only at any given light do you have less potential, but you're moving further down lower on that potential scale. So how do we translate these PI curves to actual productivity? So the first thing you do is you need to know how much light's hitting the surface, right? So you can take the daily light curves over time, and you can figure out what a composite light is over the 2-week window when we're out there sampling. But we can then look at the light extinction that we get average from all the CTD casts that we have. We can come up with the average light extinction is in this region. And then we can calculate what PAR looks like over the course of a day during the season at the different depths. You can see that there's obviously a lot of extinction. This is pretty turbid water. Close it up here so you can see that reference is roughly at peak about double what it is at the terrebone, and it's about double again from the caminata. So then if you take the light availability and you apply the PI curve for each one of our cores, you can look at what daily GPP peaks look like. So the daily integrated GPP is at taking the basic calculating GPP at every 15-minute interval over the course of the day and then integrate. So starting out with, again, just looking at what's going on in the reference sites. So you've got spring 2021, summer, fall, spring, summer, fall. It's very clear that daily GPP peaks in the summer on Ship Shoal. GPP is consistently lower in the mature dredge pit, and it tends to be intermediate between the reference and cam for the terrebone or new pit. Where's that? Okay. I should point out that the one season we have prior to dredging, that GPP in the new pit before dredging was basically the same as it was in the reference areas. So what I have now here is we have the GPP plots I showed you before, but I have respiration. But respiration is a consumption of oxygen, so that's a negative rate. And the reason why I plot them like this is so you can compare GPP to R. So what you see here is a similar pattern where we see peaks in respiration in the summer on the shoal in the reference areas. Respiration is consistently higher in the mature dredge pit than it is in the reference areas. And again, it tends to be intermediate in the terrebone. So when you combine GPP and R, it gives you the net metabolic balance or net ecosystem production for the system. And what you see in the reference areas is that during the areas where the periods of the year in which we have the highest productivity, GPP exceeds respiration. So you have a positive NEP. So that happened in both summers as well as in the spring of 2022, which is the period when we had high light as well, that we had really high light from about March through July of that year because of the way Gulf circulation was moving. But in the other periods of the year, the system was net negative, so it was net heterotrophic. When you go to the mature pit, NEP is always negative in the mature pit. And in terrebone, it's always negative, but it tends to be intermediate between the reference and cam. It makes sense if it's intermediate for GPP and for R, that NEP is probably going to be intermediate as well, right? So one thing that's really interesting about this NEP values is that the cam region, when we set our sampling, we know that hypoxia onsets on the shelf every single year, but the shoals tend to be a refuge. So typically, hypoxia, if it peaks on the overall shelf in terms of area in July, it tends to actually work its way in most years onto these shoals in sort of late August. It takes a lot longer. It's a much shallower. You've got 7 to 10 meters depth when the surrounding area is about 30. And so it takes a while for it to come on. So we've targeted our sampling before that onset, that typical max hypoxia for the shelf, to try to take that out as a variable. But one thing you do see is that the cam region is the only region where we have observed hypoxia. And we observed it in both June of 21 and 22, as well as March of 22. So that deeper pit is allowing hypoxia to form much more quickly than it does on the rest of the shoal. And the terrebone region does have lower bottom water dissolved oxygen than we typically see in the reference area, but it wasn't hypoxic during any of the windows when we sampled. So we do a lot of other characterization of the sediment in each core. We try to figure out how many syringes you can stick inside the top of a core tube and measure things like grain size, water content, bulk density, organic matter, benthic chlorophyll. We also look at algal communities and then microbes as well. I'm just going to highlight 2 of these because they're typically ones that are highly correlated with some of our different parameters. Benthic chlorophyll is often a very strong predictor of benthic primary production. Sediment organic matter content is usually one that's a pretty strong predictor of respiration. There isn't a clear differentiation between the different regions in benthic chlorophyll. There is a overall positive relationship between benthic chlorophyll and GPP within given regions, but it sort of breaks apart going across. So it's not something that's explaining the differences we've seen between the different regions. Organic matter is much, much higher in the mature pit. It makes sense. It's been dredged longer. It's had more time to accumulate organic matter. Things sort of tend to slope down into and get trapped inside the pit. There is also a trend suggesting that perhaps the organic matter content has been slowly increasing during this first year post-dredging in Terrebonne. So what do we learn about benthic prime production so far? The dredge areas exhibit lower rates of benthic prime production and higher respiration, which leads to lower NEP or more net heterotrophic. But this is not only driven by the decrease in available light from making it deeper. It's also because we're changing the GPP potential. So those PI curves have lower saturation on them. So the combination of those two lead to these lower rates of daily integrated GPP. But interestingly, the chlorophyll itself is not a good predictor of the differences we see between these regions. So what does that mean? It means that it's not just the total amount of biomass of phytoplankton or benthic microalgae is not the explainer of why we see differences in these rates, so especially in these potentials. So that's likely means that there's some sort of shift in that community. And right now we're in the process of analyzing the HPLC samples that we've collected to look at those communities. And it looks like in general, there's a shift from a diatom dominated community on the reference areas to one in which cyanobacteria and dinoflagellates may be more important in the dredge material, the dredge regions, particularly the Caminata site. And the basic, from the physiological level, they tend to have lower GPP per unit chlorophyll than do diatoms. So the suggestion is that there might be an important shift because that also is important for the food web. Diatoms are sort of a preferred food source for most heterotrophic organisms, and the cyanobacteria and dinoflagellates, even ones that are not HAB forming, tend to have be a lot less nutritious and appetizing in general. A couple other important considerations is that the dredging in these two pits was to different depths. The Caminata pit was dredged 4 meters deep to 12 meters. This new pit was only dredged 2 meters, so it's only at 10 meters. So there's something fundamentally different about that structure. The CAM is much higher in the soil organic matter. It appears that Terrebonne is increasing, but it's got a long way to go to get to that. It's also important to consider that the deeper pits experience more extensive hypoxia. And that is makes sense when you have decreases in production and increase in respiration, and you're in somewhat of a trapped basin that's 4 meters deep. So the real question here, and one of the things that we pitched to try to figure out, to try to get more funding, is what's the future of Terrebonne? Is that something that's going to progress and become something more like Caminata? Is it going to recover and become more like the reference, or is it going to stay somewhere in the middle? So that's the primary production side of things, but I wanted to give you a little bit more holistic view on things. So I'm going to present a little bit of data here on the benthic invertebrate community. This is work that's led by David Johnson and his team at VIMS. And so this is just looking at the community composition. And so we have is pie diagrams on the top. We have the reference. We have Terrebonne, the new pit, and Caminata down below. You can see that prior to dredging, which is where the red line is here, the reference in Terrebonne looked the same, dominated by an amphipod in their community. The Caminata in the spring is dominated by this small messel, the small clam. As soon as we started dredging, well, we see a drastic change, and Terrebonne looks a lot more like Caminata than it does the reference. And that sort of difference, after that immediate disturbance, the initiation of it, over time ends up with the Terrebonne ending up to look somewhat intermediate between the two areas. So a similar theme to what we're seeing in terms of the algal community. And if you look at these NMDS plots, what we have is the Caminata is in green. Over here on the left, it's very distinct from the reference and Terrebonne before dredging takes place. And if you go to the end of the study, you see that terrebone is sort of intermediate between the two. If we look at the impact of the dredging on Benthic community based on the summer of 2022, the time when we have the greatest abundance of organisms, we see that species richness in the Caminata pit is about 45% lower than we see in the reference, whereas terrebone is about 30%. The total number of individuals is about 300% greater in both of those dredge pits compared to the reference areas. We'll note that if you exclude that really small clam, it's about 100% increase in Caminata. So about 200 of the 300% is due to that clam itself. And then in biomass, in Caminata, there's actually an increase in biomass, whereas there's a decrease in terrebone. So in summary, the cam distinctly different from the reference throughout. It's dominated by a polychaete that prefers muddy sand with the annual pulses of this clam. Before dredging, terrebone looked very similar to the reference. And after dredging, the benthic community shift away from reference towards Caminata, but still in between. And it lost 80% of the biomass relative to the reference. So if you look at the conceptualization of how recovery after disturbance should look in terms of the abundance, richness, and biomass of the invertebrate communities, you have your curve here in terms of richness, you have biomass, and you have number of individuals. It's pretty clear at this point, we're right at that initial colonization community in terrebone, sampling within that first year after dredging, or actually during dredging. Dredging wasn't completed till right before our second to last sampling. And then Caminata could be in the transitional stage, or it could be in equilibrium. It's hard to tell from the amount of sampling we have. You need to look at that a little bit longer to see where we are on that trajectory. And then again, we have a very similar conclusion that we saw with the primary production. The question is what's the future for terrebone? Is it going to recover and become more like the reference? Is it going to continue to degrade, I guess is the right word, in terms of to look more like the Caminata community, or is it going to end up somewhere in the middle? So we also looked at the nekton community, and this is work that Jimmy Nelson's lab was leading, and we collected nekton each site using an otter trawl as well as a long line. And when collections were identified, measured, weighed, and sampled for tissue. Basically, when you do long lines off the Ship Shoal, you basically catch sharks and catfish. But the rest of the community is where we get the rest of from the trawl. The overall community composition, this is in spring of 21, summer of 21, and spring of 22. For the reference on the left, terrebone in the middle, and Caminata on the right. There were a significant difference between each of the fully sampled seasons. The communities overall were not significantly different in dredged and non-dredged areas. Not surprising when you got mobile organisms that aren't necessarily constrained to spending all of their time within those relatively small pits. The resource use with dredging in the summer, for pelagic representative species here, we see very similar regardless of the dredging history here in terms of benthic algae and POM as the dominant resource use, regardless of dredging history. There's a slightly less ulva when it comes to the catfish, but otherwise it's a very similar pattern between dredged and undredged as well. If you look at these hypervolumes, so what it's looking at here is sort of the spread in terms of the resource use in the isotopic space of the carbon, nitrogen, and sulfur isotopes. These clouds are kind of the hypervolume. This is basically where the different regions communities are found. What you see is that the niches in this light blue, the terrebone, are the largest in these regionally dredged areas for all of the different food sources. They're about 62% larger in terrebone versus the reference. And we see a very similar thing in terms of the catfish that are on the bottom, where they're about 400% larger in terrebone than the reference. So there are some changes in resource use, and at least the niche that's taking place in these disturbed areas, but overall the community composition of the fish communities, we're not seeing as much of a difference. I will put a caveat in there that the fish and the food web part of the study is not as far along in an analysis. We don't have that many seasons that are completed, so it's something we need to look at more closely moving forward. So the overall conclusion of this so far in these first couple years is that the impact of dredging is very obvious in terms of a reduction in primary production, an increase in respiration, and an increase in net heterotrophy, which coincide with an increased occurrences of hypoxia. That seems to impact the invertebrate communities in the sediments, where again, we're seeing that the communities in the mature dredge site looks distinct from what we see in the reference areas. But it matters a lot about the differences in the dredging, the amount of time that you're dredged. It's hard at this point for us to decouple the dredge depth from the duration since dredging. This is one of the reasons why we need a longer study. At this point, we can just tell that at 2 meters deep, 1 year after dredging, it's different than 4 meters deep of dredging 6 years after dredging. So if we can extend this study, we can start to look at when we get to the terrebone being the same age that the caminata was, we can then look at sort of whether or not how much of that is due to the dredging depth versus due to the age since dredging. It's likely to be a combination of the two. Obviously, we know we have differences in the light just simply from being deeper. So that's kind of where we're at this stage, and we're really excited that we're going to have a couple more years of funding to go out, starting again in spring of 2024, to look at this for a longer duration of time. And I want to acknowledge that this is obviously a large team effort, and this is all the folks coming off the boat after our final sampling in the fall of 2022. With that, I'll take any questions you might have. And wow, that was a lot of slides in like 35 minutes. Go ahead. No, you said that right. The reference areas are 8 meters deep. The new pit was dredged 2 meters deep to 10, and the mature pit was 2 meters further deep, so it was to 12. Because you would not be on the top of the platform of the shoal. So you would then be on to get to that depth on the shoal, you would be on the slopes. There isn't the deepest part of the shoal. Actually, you go to the western part of the shoal is actually shallower. So that area before dredging on the shoal, 8 meters, caminata was 8 meters deep beforehand. There is no 12-meter reference that's on the shoal. But it's a really good question, and you would like to do this. So when we first tried to propose this work, we actually wanted to do some transects that went off the shoal and across. So we were actually on the shelf adjacent to it and working our way up the slope. But, the budget didn't allow that.
Yeah, really nice talk. I was wondering if I was trying to remember when the big storm went over you guys, and wondering if you've looked at how big storms change these pits. Like, do they move or fill in, or have you seen any
Hurricane
effects?
Ida was, I know it very, very well, because it did $2 million of damage to our building. Was in fall of 2021. However, its path didn't go anywhere near Ship Shoal. The path, and you can tell that based upon the grain size analysis and the sediment samples that have been done with Kevin Jew's group at LSU. We can tell that there wasn't a remobilization of that. The way the path of the storm went is it came from the southeast, went through Port Fourchon, and then sat over Homa. And we're on the western edge of Terrebonne Bay. And so it wasn't in that really strong path. And it certainly was not in the storm surge in the front right quadrant at all. So that would be something that clearly you want to look at. And there's clearly other places where it would be. But yeah, we were definitely aware that we wanted to be looking at that. But when we looked at you look at all the sediment characteristics, there wasn't any change in their sediment characteristics before and after that event.
Yeah, that makes sense. Thanks.
Thanks. So I suppose this was sort of implied at the beginning of the presentation. The sediment is being taken for restoration activities up in the Mississippi, like the inshore areas. Is that correct?
Yeah, they're restoring barrier islands. Actually, when they it was, so what was this? This was summer of 2021. The governor was at so we went out to Last Island, which is the island that's somewhat famous off Louisiana because there was a hotel on this island in the 1800s and it was knocked out by a hurricane, but it's been massively degraded over time. And so there's been a massive restoration with the dredging material from here. It's 6 kilometers to the north of where the site is. And we were actually sampling there 4 days before then. I was with the governor and watching the pipe spit that sand out on that island right near there. So the naming of the pits is usually related to where that sand was going. So the Caminada was going in the area of Caminada Island. And then this is Timbalier and Terrebonne. So these were on barrier islands of right off of LUMCON that they were. So yeah.
I guess the second part of the question is there on the books to dramatically take out significant portions of these banks to supply sediment for future restoration or yeah?
Well, the challenge with these, and if you notice that on the map of the sites, these pits are really oddly shaped. And that's because there's oil and gas pipelines all over these shoals. And you can't be within a kilometer of any of the pipelines. So you end up with these very oddly shaped pits that are there. They are intending to do more barrier island restoration, which will take dredging into effect. I mean, they're clearly looking at the results from this study to figure out, hey, if we don't have as bad of an impact by only taking off the top 2 meters, is that something we should be considering with these other projects? In the end, whether or not that'll weigh enough in the decisions that we want to restore these islands and this is where the sand is, that remains to be seen. But there is, considering, that's part of the reason why they funded us to do more on this project, to try to see whether or not it stays as sort of an intermediate or recovers, because they are intending to dredge more. And it's not just here. I mean, they're dredging, they got a similar project off of Florida. They're doing some of this stuff off the Carolinas, trying to restore and it's not always barrier islands. Sometimes it's just the beach. So they do have intention to dredge more for sure. I didn't measure silica at all.
No, just kidding. So this might be buried in it looks like you took a lot of extra data, with the HPLC and whatnot. I think challenge with, in the water column, normalizing to chlorophyll is usually a way to get away from detritus, but in the sediments, it's like all detritus. So do you guys have another kind of method to disentangle what might be alive and producing? Because the GPP is definitely higher. You can see that. Is it because things are growing faster or because you just, have more live biomass and it's hidden with all this other junk?
Yeah, in the resources that we had, it's a really good question. And the resources we had available in this first round and not knowing what we were going to even find, we basically, we originally had funding just for chlorophyll, and then we had collected the extra samples and I was able to secure some additional funding to at least look at the community composition to see if we're seeing a shift in that community. But it's a real question. I mean, one of the things that happens in the pits is that they become these big resuspension zones, right? I mean, you're basically creating, part of the reasons why you end up with the organic matter there is that, the currents go over the top, the relationship between chlorophyll and phthalopigments to look and see and degrade It does look like there's more phaeopigments. There's a higher percentage of the total pigments in the caminata than there is in the reference. Haven't really run stats on it, just first glance, it kind of looks that way. But in the second phase, I'm hoping to come up with some ideas on how to better explain what is driving the differences that we see. I mean, people got other suggestions. I'm definitely open to those. I said we got till March to figure out what my plan is for the next round.
So Brian, with that organic matter and that detritus that's settling into the bottom of these pits, and so much erosion of salt marshes in Louisiana in general, do you think a lot of that marsh that's been eroded and Spartina biomass is making it to the settling in the bottom of those pits? Or
Well, when you look at the source material, when we look at the isotopes in the sediment, as well as when we take the microalgal layer on there, the sediment is not looking like the marsh. So I don't think it's really that. I do think that there is an increasing amount of river organic matter that is settling and staying there, as opposed to when it drops on top of the shelf that's more exposed, it just tends to kind of it gets deposited, and then the next current, it moves it off. And then you have a lot of sort of the buildup just when you're in that kind of a pit, you just tend to accumulate organic matter over time. But at this point, at least, it doesn't look like that it's more of a salt marsh signal in that pit.
okay. One more question. How long have these shoals been there?
The shoals, well, I mean, they were bare islands. They basically went, depending on what shoal you're talking about, most of them went below the water surface in the last 100 to 150 years.
Okay.
So I'm going to follow up on Charlie's question because I was waiting for you to say isotope and I got all tingly. So I wondered, have you thought about looking at the compound specific isotopes or specifically at the inalkanes and seeing if you can parse some of that out by like the ratios of long and short chain alkanes to get at some of those questions?
I have thought about it. Unless you got a big bag of money somewhere over else, like I don't really think we're going to get to that with here. We just had the site visit from BOEM last month, and we did talk about potential ways to expand and try to increase our understanding. And they haven't ruled out the possibility of providing some additional resources in the future. So we'll see if we can convince them of some of these things. I mean, it's a good idea. It comes down to the prioritization of which things that they want to really focus on and how much they want to know the true answer, or they just want to be able to know what the impact is. And that's unclear. And, but it's a good idea, and it is something we've thought about, but trying to figure out how to parse up the limited resources as far as you can go. We all fight that battle, right? So.
Notice in the recovering pits, the amphioxus didn't seem to come back. Is that significant biologically in that area, or what do you attribute to that lack of recovery in that population?
Yeah, I think it is significant that it hasn't come back. Because it's, when we're talking about the food sources for the next levels of the food web, I think it is important that we're seeing that shift in that community, understanding exactly what the mechanism for it not coming back. I'm not really sure yet. I mean, we haven't at this point. What I've basically given you is a snapshot of the work that my lab's done, the stuff that David's lab's done, and where Jimmy's at. And we're at the stage where we're trying to kind of put the pieces all together. But it's clear that that's a shift, and that was obviously a central part of the food web. So I think it's important, but how important relative to some of the other shifts, I'm not really sure yet. Oh. I knew she was on ball, but you
Yeah.
told me to look, so I looked.
I was wondering, have you looked at differences in like resource quality between the sites, like I guess with respect to detrital quality as far as like recalcitrant versus labile or anything like that?
No, I mean, again, it's like we're kind of just unfolding a lot of these results right now. I mean, so those are things that, it's not something that, I've thought about, but, in terms of as a potential important thing to understand. But in terms of trying to figure out what it is that we were going to do, and the other thing you got to remember is that with BOEM, there's a cooperative agreement and they want you to do this, 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 and this. And there's a bunch of lomnyap that I've put in here that wasn't part of that list, but you can only go so far down that list because of that. For example, the water column wasn't even part of any of this, but I'm really intrigued, and I've got, one of the things that I'm really trying to do in my new postdoc on this project is what we're doing, because we have now these great PI curves and we have them, how they vary over the different seasons. We have lots of information on light extinction. There is some great remote sensing data out there on watercolor, and we've been able to show pretty good relationships between our total suspended sediments and our light extinction parameters. And so the idea is that we can model on sort of the daily time step. That's the goal, of course. They're going to have gaps in the data, but over the year, sort of what does productivity look like? Because when you take these snapshots, are you hitting a high point or a low point? Like it's hard to assess annual rates out of these three windows. And so I think that's important to understand some of these dynamics. And then that will also help us inform what some of the directed additional lanyap type things we're going to focus on. You guys have all had all these great ideas about things, some of which we thought about more than others. And, we'll probably try to do some of them, but it's trying to figure out what those priorities are as you're moving forward. And a lot of this, those of you who haven't had funding with an organization like BOEM, there's a lot of give and take. Like NSF, you get an NSF grant, and they basically fund you and you turn in your annual report. They give you a couple of questions sometimes on what you spend in your annual report, but as long as you're producing, it's okay. For some of these other ones, it's like they have a specific thing that you're tasked to do, and you've got to give them the data for each of these things that you're tasked to do. And so that's always where your focus is.
Last question.
Yeah, just a more general sort of question. You mentioned these only submerged maybe 150 years ago. I've got no idea of the sort of evolution of a barrier island after it submerges, but I just started thinking about if you start digging it, has anyone cored these banks? And deep cored them? And one of the questions I'm just sort of thinking is some of that organic matter in the pits actually buried organic matter that is now being exposed and is coming out of the pits?
Now that is something that I've actually thought about a lot, and is actually something that I'm trying to convince them to give us some extra money to do. Because that's actually something I'm really interested in. As you're eroding away, you're eroding away. These were bare islands, right? And most of the bare islands, you got your beach side and you got your vegetated side on the bay side of things. And so that those tend to be pretty rich in organic matter. And as they sort of eroded away, at some point, you're going to get to where those layers are and things. And so I am interested in that. I'm not aware of any studies that have really done that sort of deeper core through sand shoals. If somebody knows about somebody that did, please let me know, because I would be interested in that. But it is something we have talked about, because that is the question. What is the source of the organic matter? Is it simply just an increase in the amount of trapping over time that you build that up? Or, is that some other source? That leads into sort of, okay, if it's not what Charlie was asking about, that's the eroded wetland material coming out. In some respects, it could be some of that, because that is part of what gets resuspended. Part of the high turbidity we have in the river plume is stuff that is derived from land, right? So, but in terms of isotopically at this point, with the coarseness at which you can measure those isotopes, we're not detecting it being a signal that is similar to what it is in terms of organic matter on the marsh. It looks similar to what you see in the other sand.